Oh, look! It's Mini-Me! This one is a little undercooked.
Oi, oi, you muddy fugglers! Get in here, sit down, we're going to read some news. Decades ago, I read a book by a chap called Eric Drexler called The Engines of Creation, and his argument for the trajectory of nanotechnology was that we would be starting from the ground up, from proteins and enzymes and the inner machinery of cells, and working upwards from there to create functional nanoscopic machines. Which, if we're looking at mRNA technology and all the stuff that came out during the pandemic, that seems to be the direction things have been going. Well, there's only a matter of time until they're able to actually create a fully functioning cell, and along that lines, this article on the Daily Mail, written by that good old chap William Hunter. Hello, sir. Scientists build a cell from scratch. Synthetic organisms can feed, grow, copy its DNA and divide in world-first breakthrough. Scientists have built a synthetic cell from scratch in a world-first breakthrough that paves the way for creating entirely artificial forms of life. Tiny blobs, dubbed Spud cells, are about 50 times smaller than a normal bacterium and are made of microscopic water droplets encased in a fatty membrane. That bubble is filled with enzymes, chemicals, and small snippets of DNA that allow Spud cell to perform some of the basic functions of life. According to its creators, the synthetic cell can now feed, grow, copy its DNA, divide, and even change over generations in a process resembling evolution. This is not the first time that scientists have attempted to make synthetic life, but unlike those earlier attempts, Spud cell is entirely constructed from the ground up. Scientists hope these cells could one day revolutionize medicine by acting like mini biological factories, pumping out medicines and other chemicals. Lead author Professor Kate Amidala of the University of Minnesota Twin Cities says we've replicated in chemistry what only used to be possible in biology, the complete set of behaviors of a cell. It proves that the most fundamental functions of life, like growth and replication, do not need a mysterious magical spark. Rather than breaking down and reconstructing existing cells, these almost living bubbles are made from nothing but artificial chemicals. Like all living organisms, Spud cell contains stretches of DNA that contain the instructions for making all the proteins the cell needs to survive. Spud cell also contains a sort of biochemical toolkit called Pure, which includes everything the cell needs to turn those DNA instructions into proteins. While a human genome contains around 3 billion pairs of DNA, Spud cell contains just 90,000. Still kind of a large number, I don't know. Is that small? Biologists had previously speculated that the smallest genome a living cell could have is 113,000 pairs, making this synthetic organism much smaller than the theoretical limit. This means that Spud cell is far simpler and much less sophisticated than even the most basic life form. However, despite the simplicity of its genetic code, these fatty bubbles are still able to replicate several basic functions of life. Spud cell can feed by fusing with minuscule feeder liposomes, hollow spheres made of fatty lipids which contain all the nutrients the cell needs to function. Its DNA contains instructions to use that food to create replicas of its genetic code, preparing it for reproduction. The cells can even undergo a basic form of division by flooding their membrane with a protein that creates a repelling force, essentially tearing the cell apart at the seams. Most impressively, the cells even appear to be capable of a form of natural selection over multiple generations. In an experiment published in a preprint paper, the scientists gave some Spud cells a mutation that helped them gather more food and grow faster. After 5 generations, these mutated cells had outcompeted their rivals, and 60% of genomes contained the mutation. To develop the concept further, Professor Amidala and her co-authors have founded a public benefit research institute named Biotic. However, Professor Amidala cautions this does not mean Spud cells are alive. The process of selection can't be considered evolution because the researchers had to insert the mutation from outset rather than having it arise naturally. I suppose it would have to have many generations and potential mutations for there to be actual evolution. Spud cells also can't naturally divide over many generations, and the researchers had to press them through a membrane with tiny holes to get multiple rounds of division. So you're basically squeezing them through tiny holes like Play-Doh and hoping that whatever pops out the other end still works. This process is also incredibly crude compared to the division that happens in real cells. Since the Spud cells don't tear themselves apart evenly, they don't always get the right number of genomes in the finished offspring. After 5 division cycles, the researchers found that only 30% of cells still carried the full genome. Professor John Dupree, a philosopher and founder of the Center for the Study of Life Sciences at the University of Exeter, told the Daily Mail, "This work is undoubtedly technically very impressive. Whether it will ultimately underlie diverse applications across all of biotechnology is more questionable. Even assuming the synthetic biology will eventually produce entities with all the capabilities of a living bacterial cell, it is doubtful whether this will ever be a more efficient technology than modification of naturally evolving cells." Scientists have also criticized the paper's publication, which was released to the public without peer review after being reportedly rejected by the journal Cell. Professor Kirshen Gottfried, a molecular biologist at Heidelberg University, told the Daily Mail, "History has shown multiple times that press before peer review can go wrong. A good ethical standard would be to refrain from reporting until the paper has gone through the normal peer review procedure. Life on Earth may have started thanks to a modified version of a modern-day RNA. Life on Earth may have started thanks to a modified version of modern-day DNA sister molecule, scientists believe. Could it not be a cousin molecule or a brother molecule? DNA is the backbone of life, and almost all of our planet depends on it. But on primordial Earth, a primitive version of its lesser-known sister RNA was the focal point of evolution, experts say. RNA is structurally similar to DNA, except one of the 4 fundamental pieces, thymine, is substituted for uracil. This changes the shape and structure of the molecule, and researchers have long believed this chemical was vital to the development of Earth's first life forms. An accidental discovery by Harvard academics published in December 2018 found that a slightly different version of RNA may have been the key ingredient allowing life on Earth to blossom. Scientists claim that a chemical called iosine may have been present in place of guanine, allowing for life to develop. This slight change to the bases known as nucleotides may provide the first known proof of the RNA world hypothesis, a theory which claims RNA was integral to primitive life forms. It's not quite the same subject as what we're reading, but thank you for adding that. What are the 7 signs of life? Cellular organization. Well, I mean, if you're a single-celled creature, then no. The ability to reproduce, yes. Growth and development, yes. Energy use, yes. Homeostasis, yes. Basically keeping your internal environment as stable as possible in contrast to the external environment. Responses to their environment, okay, yes. Ability to adapt, which is kind of like sex, is it not? Well, this is interesting. This is a step towards entirely artificial life. So the other route is being able to genetically engineer existing cells to do what you want them to do. And this is effectively trying to build a cell from the ground up to arrive at complexity from a collection of more simple parts. So I don't know, is it more than just a experiment on fleshing out whatever tech that they have to see if they can throw stuff together that works? Because it's not going to be as efficient as having a cell and engineering an existing bacterium to do what you want it to do. But maybe there are future benefits that I am not aware of. But I imagine taking a fully functioning entity and modifying its DNA is going to produce a more robust result than what they're doing here. But this is an interesting illustration of the sophistication of all of the different sciences that are reverse engineering the cells of our bodies, and their ability now to throw together all those different disciplines to create something that is lifelike is pretty mind-blowing. Blows my bloody mind. And where are we? 2026. Crazy shit is coming down the pipe, and this is only going to get more sophisticated, so look away at your peril. Keep looking, because we need as many eyes on this as possible so we know what's going on. So yeah, Nanotechnology through biology is coming. It's here, and it's going to be even more here soon. And they're getting well clever about it. Thank you for joining me wherever you are. Hit the like button if you like what I do. But that is it for now. And the only thing I have left to say is Pigwig out.
Oink, oinkity, snort, snuffle, snuffle, squee, oink, oink, wee, squeal, snort, snort, oinkity, squee. Oink, oink, oinkity, squee, squee, wee, 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 snuffle, snort, squeal, squeal, oinkity, oink, snort, snort, squee, oink, 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 wee, 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 oh. Snort, snuffle, oink, squee, squeal, wee, snort, oinkity, oinkity, oink, oink, snuffle, wee, squee, snort, oink, squeal. Oink, oink, oinkity, squee, squee, wee, 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 snuffle, snort, squeal, squeal, oinkity, oink, snort, snort, squee, oink, 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 wee, 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 oh. Oink, oinkity, snort, snuffle, snuffle, squee, oinkity, oink, oink, snuffle, wee, squee, snort, oink, squeal. Oink, oink, oinkity, squee, squee, wee, 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 snuffle, snort, squeal, squeal, oinkity, oink, snort, snort, squee, oink, 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 wee, 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 oh. Squeal, squeal. Snort, shuffle, oink, re-squeal, re-snort, oinky, oinkity, oink, 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 snuffle, re-squeeze, snort, oink, squeal, oink, oink, oinkity, squeeze, squee, re-re-re, snuffle, snort, squeal, squeal, oinky, oink, snort, snort, squee, oink, oink, oink,